Today, I will talk about the impact of microbial biofilm in healthcare associated setting. The contents of the lecture include the general formation of biofilm, types of microorganisms involved in the formation of biofilm, then the microorganisms which are most frequently associated in hospital and healthcare system, detection and diagnosis of microbial biofilm in hospital environment, and finally the preventive measures for controlling the microbial biofilm in hospital environment. Biofilm formation has become a significant problem in healthcare industry. They may be formed on living or non-living surfaces and can be prevalent in natural, industrial, and hospital settings. Biofilms are the communities of microorganisms that are attached to surface and play a significant role in the persistence of bacterial infections. Biofilm have varied morphologies depending on the constituent of bacteria as well as the condition under which that biofilms are formed. Commonly, the biofilms are involved in a multitude of serious chronic infections. They are formed by a spectrum of microorganisms, including pathogens, and they provide a means for these organisms to protect themselves against antimicrobial agents. There are several mechanisms have been proposed to explain this phenomenon of resistance within the biofilm. The biofilm formation are one of a major global challenge to control infection and healthcare associated infection owing to their inherent tolerance and resistance to antimicrobial treatment. The biofilms have been shown to develop on medical device surfaces, and dispersal of single and clustered cells indicates a major risk of biofilm dissemination within the host and increase the risk of infection. I already told in the previous lecture, the biofilms are composed of extracellular polymeric substances. They are simply called as EPS. The EPS is consists of three major constituents such as the exopolysaccharide, protein, and nucleic acid. The structure of the biofilm is made up of two regions. One is hydrophobic region and hydrophilic region. Hydrophobic is nothing but the region does not like water. Then hydrophilic means the component that likes water. Also, it can be easily soluble in the presence of water. Among these two, hydrophilic region is a major component which is highly present in the microbial biofilm. The biofilm matrix encloses the bacterial cell within it and facilitate the communication among them through a biochemical signal as well as a gene exchange. The EPS matrix also traps extracellular enzymes and keeps them in close proximity of the cells. Also, some biofilms have been found to contain water channels that help to distribute nutrients and signaling molecule. In addition, it was observed that an extracellular DNA is a main structural component of many different microbial biofilm. Then the enzymatic degradation of this extracellular DNA can weaken the biofilm structure and release the microbial cell from the surface of the matrix. Generally, the biofilms are formed by bacteria which colonize plant, especially Pseudomonas fluorescens and Pseudomonas putida. Also, some related Pseudomonas species that are usually plant associated bacteria that are commonly found on plant root, leaves, and soil. Many different bacteria forms biofilm, including gram-positive, especially the Bacillus species, Listeria monocytogenes, and Staphylococcus species. Also, some lactic acid bacteria such as Lactobacillus plantarum and Lactobacillus lacti. The gram-negative bacteria include Escherichia coli and Pseudomonas aeruginosa. Also, some cyanobacteria can able to form biofilm in aquatic environment. The biofilm formation is commonly considered to occur in three main steps. One is attachment to a surface, second one is maturation, and third one is detachment, which is also called as dispersal. The biofilms develop via initial attachment, which depends on transport of the bacteria to a surface, which is passive in the case of non-motile bacteria, for example Staphylococcus, and active in the case of motile bacteria, especially the Pseudomonas aeruginosa. The interactions of motility appendages with fluid and surfaces may promote motility, attachment, and dispersal of bacteria on surfaces prior to biofilm formation. Let us see the first thing, the attachment of bacteria on biofilm. Attachment itself governed by specific protein-protein interaction of bacterial surface with human matrix protein. That means the subsequent steps do not differ in principle between motile and non-motile bacteria. They involve proliferation, embedding in an extracellular matrix, and maturation. The strong production of surfactant are controlled by quorum sensing that leads to biofilm detachment. The biofilm attachment to an abiotic surface such as a catheter mainly depends on the bacterial surface hydrophobicity, but this mechanism is believed to have minor importance. The extracellular DNA has an important role in biofilm formation. Then matrix formation. After attachment to tissue or matrix covered devices is accomplished, infectious bacterial biofilm may grow by proliferation and production of an extracellular matrix. The functions of the matrix is to provide adhesion between bacterial cell, thereby enabling the formation of a multi-layered biofilm. The in vitro evidence indicates that the biofilm matrix consists of a multitude of components of different chemical nature, including exopolysaccharide, protein, and other polymers. These components may also facilitate the formation of bacterial agglomeration that do not necessarily constitute a biofilm and provide protection from antibiotics and host defense independently of biofilm formation. Teichoic acid is a major cellular component commonly found in gram-positive bacteria. They are negatively charged and have been shown to contribute the formation of biofilm, especially the gram-positive bacteria Staphylococcus. Most likely, they interact with other surface polymers and function as a scaffold for protein attachment. Next, the third one, dispersal or detachment of biofilm. Dispersal of cells from the biofilm colony is an essential stage of biofilm life cycle. Dispersal enables biofilm to spread and colonize new surfaces. Enzymes that degrade the biofilm extracellular matrix, especially the dispersin B and deoxyribonuclease, they may play a role in biofilm dispersal. Biofilm matrix degrading enzyme may be useful in anti-biofilm agent, and some recent evidences has shown that a fatty acid messenger, especially cis-2-deconic acid, is capable of including dispersion and inhibiting the growth of biofilm colonies. Also, nitric oxide has shown to be trigger the dispersal of biofilm of several bacterial species. This nitric oxide has the potential for the treatment of patients that suffer from chronic infection which is caused by the biofilm. Next, the significance of biofilm in healthcare system. Biofilms are of more importance in infection control and healthcare associated infection owing to their inherent tolerance and resistance to antimicrobial therapies. Biofilms have been shown to develop on medical device surfaces, and dispersal of single and cluster cells implies a significant risk of microbial dissemination within the host and increased risk of infection in healthcare associated setting, hospitals, or in a patient home. Some of the microorganisms most frequently associated with healthcare associated infections include gram-positive bacteria, especially the Staphylococcus aureus, Staphylococcus epidermidis, Enterococcus faecalis, and gram-positive bacteria include E. coli, Klebsiella pneumoniae, Proteus mirabilis, Pseudomonas aeruginosa, and some of the yeast, especially the Candida species. These bacteria are commonly found and forming a biofilm in many medical devices. For example, on the surface of central venous catheter, the organisms such as coagulase negative Staphylococcus, Staphylococcus aureus, Enterococcus faecalis, Klebsiella pneumoniae, Pseudomonas aeruginosa, and Candida albicans. These are able to form the biofilm on the central venous catheter. Then the organisms such as viridin streptococcus, coagulase negative staphylococci, enterococci, and Staphylococcus aureus, they are able to form the biofilm on prosthetic heart valve. Next, urinary catheter surfaces are accomplished by Staphylococcus epidermidis, Escherichia coli, Klebsiella pneumoniae, Enterococcus faecalis, and Proteus mirabilis. Then artificial hip prostheses are contaminated by coagulase negative staphylococci, beta hemolytic streptococci, enterococci, Proteus mirabilis, bacterioid Staphylococcus aureus, viridin streptococci, Escherichia coli, and Pseudomonas aeruginosa. Then the pathogenic organism forming biofilm on artificial voice prosthesis are Candida albicans, Streptococcus mitis, Streptococcus salivarius, Candida tropicalis, Streptococcus subrenus, and Staphylococcus epidermidis. Next, the intrauterine devices are contaminated by Staphylococcus epidermidis, Corynebacterium species, Staphylococcus aureus, Micrococcus species, Lactobacillus plantarum, Group B Streptococci, Enterococci species, and Candida albicans. These are some major pathogenic microorganisms which are commonly forming biofilm on medical devices. Commonly, the initial contamination of the medical devices most likely occurs from a small number of microorganisms, which are often transferred to the device in question via the patient or healthcare worker's skin, contaminated water, or other some external environmental sources. Also, a wide range of microorganisms has been implicated in medical device related infection, especially the Staphylococcus epidermidis and Staphylococcus aureus are the most commonly associated with biofilms formed on medical devices. In addition to staphylococcal species, the identification of multi-drug resistant gram-negative bacteria, particularly Aeromonas species, Escherichia coli, Klebsiella pneumoniae, and Pseudomonas aeruginosa are becoming more widespread in long-term care facilities and acute care hospitals. Next, we will see how the microbial biofilms are formed in medical devices. First, we will discuss central venous catheter. Central venous catheters are utilized to deliver fluid, blood product, nutritional solution or medications and for access in dialysis treatment. Both of the outer part of the catheter and the catheter lumen can become contaminated. So, it can be resulting in the formation of biofilm. Within the first week of catheterization, extraluminary biofilm is considered the major cause of catheter-associated bloodstream infections. Therefore, the patient who require long-term use of such devices for intravenous access, such as bone marrow transplant patient, are at a greater risk of bloodstream infection. It also been noted that catheter colonization and biofilm formation on central venous catheters occurs very easily compared to other ways. The microbial colonization and biofilm formation occurred as early as one day after catheter insection in a cohort of adult cancer patients whose central venous catheter were removed. Next, urinary catheters. Urinary catheters are tubular latex or silicone devices that are commonly used to measure urine output. Also, collecting urine sample during surgery, prevent urine retention or control urinary incontinence. For patient undergoing catheterization, the risk of developing a catheter-associated infection increases by approximately 10% each day the catheter is in place. The biofilms formed by the pathogenic microorganism can readily develop on both the inner and outer surfaces of the urinary catheter, and the ascending colonization cannot be avoided solely through the hygiene measures. Therefore, in order to prevent such infection, it is important for doctors to utilize catheters only when necessary and to avoid catheterization for extended period of time. The contaminated bacteria may originate from those organisms that colonize the periurethral skin, which migrate into the bladder via the mucoid layer that forms between the epithelial surface or the urethra and the catheter. Also, the contamination of the urine within the catheter can also be a source of bacteria, then go on to cause infection. Next, ventilator associated pneumonia and endotracheal tube. Ventilator associated pneumonia has been reported to be prevalent after 48 to 72 hours in a patient who have been incubated and are on mechanical ventilation. The ventilator associated pneumonia has a major implications for both the patient and the healthcare system. This leading to prolonged hospital stay and increased healthcare cost. The risk of developing ventilator associated pneumonia following incubation with mechanical ventilation is increased 6 to 20 fold, with mortality rate ranging from 24 to 76%, significantly higher than mortality rate for urinary tract and skin infections. Then the endotracheal tube have been reported to be a factor in the acquisition of ventilator associated pneumonia. When compared to other devices, the endotracheal tubes are easily contaminated by the biofilm, the microorganism that have been documented to colonize the endotracheal tubes, including multi-drug resistant bacterium such as methicillin resistant Staphylococcus aureus and gram-negative bacilli such as Klebsiella pneumoniae, Escherichia coli, Pseudomonas aeruginosa, and some Acinetobacter species. Next, surgical site infection. Surgical site infection, nothing but the wound infection that occur following a surgical procedure. They can occur due to the contamination of a wound by microorganism derived from the patient own skin. The most frequent microorganism associated with surgical site infection is Staphylococcus aureus. It is commonly found in the skin as a normal flora. Also, the growth of microorganism within a biofilm has been associated with a number of chronic infections. For example, Pseudomonas aeruginosa forms biofilm in the lungs of a patient with cystic fibrosis, and despite the egregious use of antibiotics, the microbial colonization is often a lifelong problem that leading to chronic inflammation and lung tissue damage. The biofilm forming Pseudomonas aeruginosa also has a role to play in the persistence of cutaneous wound infections and has been shown to form biofilm in both human and veterinary wound. Then we will discuss how to detect and diagnosis of medical biofilm. Till now, there is no detection method is available for the diagnosis of a biofilm within a clinical setting. The use of traditional culture method to determine colonization is not indicative of biofilm growth. Furthermore, negative results from swab sample may not necessarily imply the absence of an infection, but could possibly be due to the slow growth rate within a biofilm of species that cannot be detected within usual detection range. One of the barriers that can make successful diagnosis difficult is the emergence of small colony variants. Small colony variants are a subpopulation of biofilm bacteria that produce small colonies, develop resistance to antimicrobial action, and can evade detection owing to their slow growth rate. For example, patients often develop a gradual onset of joint pain without the clinical sign and symptoms of infection, such as fever, surgical site wound drainage, redness, or swelling. In this case, Staphylococcus infections are of particular interest in terms of the problem associated with small colony variants. So, in order to improve the diagnosis of device-related infection, methods such as the sonification of suspected infected implants may improve culture positivity. In addition to this, more sophisticated molecular methods of identification, including PCR and fluorescence in situ hybridization, are now being used to identify bacteria in complex biological samples and are proving to be a more accurate means of detection. Next, the control measures of biofilm in medical devices. Because of the harmful impact of biofilm for the individual as well as the healthcare facility, there are many research is being conducted to figure out how to fight biofilm related infections. There are three strategies to fight these infections, including inhibiting bacterial cells from binding to the surface, interfering with biofilm development, and killing the bacteria within a biofilm either directly or through the disaggregation of the biofilm matrix. Inhibiting bacteria from binding to a device can be done by creating a surface that the bacteria cannot physically bind to, or by coating the surface with antimicrobial agent. For example, the silicone used for medical devices are coated with some materials containing ester and cyclic hydrocarbon moieties, which showed 96.7% reduction of the surface area with attached bacteria when compared to the silicon coated with silver hydrogel coating. One of the most important point about using a material that inherently block bacterial binding rather than one coated in antimicrobial agent is that there is no selective pressure for organism to develop antimicrobial resistance. Next, quorum sensing, which involves the communication between cells using autoinducers. That means the small diffusible signal molecule. It is an important factor in the creation of biofilm. The use of molecule to inhibit the signal generator, the signal molecule, or the signal receptor is an important step in the prevention of biofilm formation. Then, in order to effectively kill the bacteria in a biofilm, many times a combination of different antibiotics along with biofilm matrix degrading substances must be used. Particularly, the degradation of the biofilm matrix is important in order to expose all of the sessile microbial cell to both the host immune defense as well as the antibiotics. Dispersin B is an important molecule degrade the biofilms of many bacteria including Staphylococcus epidermidis. In this way, the microbial biofilms are controlled on the medical devices which are commonly employed in hospital as well as the healthcare settings. Thank you.